This is part 3 of the chapter 3 lecture notes. The last thing that we were going over was vesicular transport, which means that materials, it's a large amount of materials, which is too large to cross over the plasma membrane through diffusion or like through a carrier or channel protein. So vesicles have to be used, or sometimes called transport vesicles. If the material is being moved into the cell, we call it endocytosis, and if it's being moved out, we call it exocytosis. These processes do require energy, so they're an example of active transport. And then for endocytosis, we had the last thing that we talked about was that there are 3 major types, and we looked at a slide that shows receptor-mediated endocytosis. And here is that slide. So basically what you have is, here's your plasma membrane. As you can see, all along here is the plasma membrane of the cell. The brown area or beige area represents the inside of the cell. So these little purple molecules are going to be some molecule like, for example, cholesterol. The LDL, or low density lipoprotein cholesterol, which we also know that as the bad cholesterol, is moved into our cells this way. And what happens is the molecules called ligands, the cholesterol would be an example of a ligand, binds to the receptors on the plasma membrane, and then the plasma membrane in step 2 forms an indention, and it eventually will pinch off and form a vesicle, a coated vesicle coated with those receptors. So the 4th step would be a lysosome would come in and fuse with that vesicle and release digestive enzymes, which would help to break down the ligands, in this case the cholesterol. And then the vesicle would move back up to the plasma membrane and fuse with it. And so the plasma membrane doesn't get smaller, it just stays the same size, because as a vesicle pinches off, another one comes in and fuses back with it. So the receptor-mediated endocytosis is one type. Then we have pinocytosis, which is another word for that. Sometimes we call it cell drinking, because the material that's being moved inside the cell is liquid. It's a solution. So when it's a solution, which is more of a liquid, we call it cell drinking or pinocytosis. And then if it is a solid, a large amount of solid, we call it cell eating or phagocytosis. An example of that would be when our white blood cells engulf a bacterium. So here's an example of that. This is the plasma membrane of our white blood cell. Here's the bacterium, and notice that we're seeing a vesicle form in the plasma membrane, and eventually pinches off and brings that bacterium inside our cells. Lysosomes come and fuse with that vesicle and break down the bacterium, and then eventually the vesicle will fuse with the plasma membrane again at a different place, and it will release the materials outside the cell, and that process is exocytosis. So exocytosis is the reverse of endocytosis. This table is a really good table for giving you a good summary of the transport processes. And it's continued on here, but it shows you diffusion, osmosis, facilitated diffusion, active transport, and then the types of vesicular transport. All right, what is the difference between active and passive transport processes? Well, the easy thing is that active processes require energy. Mostly that energy is in the form of A. That's an A. T. ATP, or adenosine triphosphate. And passive transport processes don't require that. But if you wanted to be even more specific or more detailed, active transport processes move substances against their concentration gradient. They move the substances from low to high concentration, whereas passive processes move substances from high to low. All right, number 12. During digestion, the concentration of hydrogen ions in the stomach rises to many times that within the cells lining the stomach, where hydrogen ions are produced. Is the type of transport passive or active? So if the cells lining the stomach produce the hydrogen ions, here's a cell lining the stomach, and then here's an H plus inside that cell. Sorry. Okay, but outside the cell, you're just building up more and more and more hydrogen ions. Then the concentration outside the cell really is high, and the concentration inside the cell is low. So hydrogen ions are being moved from their low to their high concentration, and that is an active process. We also can call that pumping. We're pumping hydrogen ions out of the cells into the stomach. All right, 13, when certain types of white blood cells encounter bacteria, they are able to engulf them and bring them into the cell. This process is called phagocytosis, which is a type of endocytosis. All right, you do need to know the difference between the terms cytoplasm and cytosol. Cytoplasm is the general term for material between the plasma membrane and the nuclear membrane, whereas the cytoplasm contains the cytosol, which is the fluid, and all of the organelles. So the difference is cytoplasm includes the organelles, and cytosol does not. Cytosol just means the fluid inside of the cytoplasm. Cytosol differs from extracellular fluid in that it contains higher levels of potassium ions and lower levels of sodium ions. It also is going to contain higher levels of dissolved proteins than what you would find in the extracellular fluid. So the cytosol is thicker than the extracellular fluid. Organelles are small organs. You can look at them that way. They're little organs that reside inside of our cells and perform certain functions. The cytoskeleton is composed of protein fibers, and these fibers are either called microfilaments, intermediate filaments, or microtubules. You can see intermediate filaments are in the structure are purple. They're shown by these purple filaments. The microtubules are thicker than that, and the microfilaments are much thinner. So the microfilaments are the thinnest. They're usually composed of a protein called actin, which is also helps in muscle contraction. And what they do, they attach the plasma membrane to the underlying cytoplasm. So this is the function of the microfilaments right here. Okay, and they do participate in muscle contraction in muscle cells. Intermediate filaments are intermediate in size. They strengthen and stabilize the cell. And then microtubules are the largest. They are hollow tubes. They give the cells strength and rigidity. They anchor organelles in place. And they do something very special down here at the bottom. They form the spindle apparatus during cell division, which we will talk about in a little bit. Microvilli are areas of the plasma membrane where the plasma membrane has been folded, and it forms these finger-like projections, and they have a special role. They increase the surface area of the membrane. So, for example, cells that need to absorb a lot of nutrients, the cells that line our small intestines, for example, or our intestines, are going to be covered with microvilli because they can absorb a lot more nutrients that way. Centrioles are structures that are important in cell division. The centrioles are composed of microtubules, and they produce the spindle fibers that help to move the chromosomes or separate the chromosomes and move them to, they produce the spindle fibers that help to separate chromosomes and move them to opposite sides of the cell during cell division. So centrioles are very important during cell division, and they are composed of microtubules. Cilia they look a little bit like microvilli, but they're much longer. They're little hairs that line the surface of some cells. For example, they line the surface of our respiratory tract, the cells that line our respiratory tract, and they help to move mucus and trapped particles up and out of our respiratory tract. Flagella are more like long tails, like just think of a sperm. Ribosomes are organelles, but they are not enclosed with a membrane. So ribosomes help to manufacture or synthesize proteins. That's their job. Some of the ribosomes float in the cytoplasm, and they're called free ribosomes. And then some are attached. We call them fixed ribosomes. They're attached to the endoplasmic reticulum. And that means the endoplasmic reticulum with ribosomes attached is called rough ER, rough endoplasmic reticulum. Proteasomes break down proteins. They contain proteases, which are enzymes that help to break down proteins. The endoplasmic reticulum is continuous with the nuclear envelope. So you have a nucleus inside the cell, and it's got endoplasmic reticulum attached to it. And the endoplasmic reticulum forms these channels and chambers called cisternae. There are two types. There's the smooth ER and the rough ER. And you can see in the picture, the rough ER is closest to the nucleus, and it contains ribosomes, and the smooth ER does not. Since the rough ER contains ribosomes, it is more involved in modifying and packaging newly synthesized proteins, and the smooth ER is more involved in synthesizing lipids and carbohydrates. The main function of the endoplasmic reticulum is the synthesis of the chemicals protein, carbohydrate, and lipid, the storage of materials, isolating them from the cytosol. In our muscles, the endoplasmic reticulum stores calcium ions, transporting materials through the cell, and detoxification of drugs. So the cells in our liver are going to have a lot of endoplasmic reticulum. Smooth endoplasmic reticulum is called smooth because it has no ribosomes. It does synthesize lipids and carbohydrates, particularly phospholipids, which form the plasma membrane, and the cholesterol that's also found within the phospholipid bilayer. Steroid hormones, which are derived from cholesterol. Glycerides, especially triglycerides, which we talked about, are lipids that form body fat. And glycogen, which is the storage form of glucose in skeletal muscle and liver cells. Glycogen is also known as animal starch. It's just like starch, but it's found in animal cells instead of plant. Rough endoplasmic reticulum has ribosomes, so it's going to be more involved in protein synthesis. The Golgi apparatus takes the chemicals that are produced in the rough ER and modifies, and smooth ER modifies and packages them within vesicles. The Golgi apparatus forms lysosomes. It's the last step in the formation of a lysosome. A lysosome is a vesicle that's filled with digestive enzymes, and it helps to digest chemicals.